Hello students, this is Tanika. Welcome to my channel PrepBiology. So this is part 2nd of class 14th. In part 1 of class 14th, we have started post fertilization events, and in that class we have discussed about the endosperm and embryo. In this class, we are going to discuss about the formation of seed and formation of fruit. Let's start with the lecture. Now, we know that under the post-fertilization events, the events which occurs after the fertilization, there are 4 events occurs. Okay, so in the previous class we have discussed about the endosperm, its functions, its structure. Embryo, we have discussed the monocot embryo and the dicot embryo, its structure. Now in this class we are going to discuss about the seed, their structure, their types, and about the fruit, structure and types. Okay, so let's start with the seed. We know that seed is the final product of the sexual reproduction. Okay, so in the final product of the sexual reproduction is the formation of seed, and the formation of seed is from the maturity. When the ovule gets mature, when the ovule gets mature, the formation of seed occurs. Okay, so the seed is formed from the ovule. Now, seed consists of mainly 3 parts. First one is the seed coat, second is the cotyledons, and third is the embryo axis. So if we talk about the seed coat, in the seed there are special 2 protective layers or the seed coats. First one is the testa, second is the tegmen. Okay, these form the seed coat. Okay, if we see the testa and the tegmen, if we see this structure, look at this structure. This is the structure of the seed, any seed. So the testa is the outermost covering, protective covering of the seed. Testa is the outermost protective covering of the seed, and inner to testa, this is the tegmen present, and tegmen is the inner protective covering of the seed. Okay, testa forms the outer protective covering, tegmen form the inner protective covering. Now, if we see some features of each layer, the testa is smooth in nature and it is thin. Okay, and the tegmen is thick, somewhat thicker than the testa, and the testa is derived from the outer integument of the ovule. Okay, so we know that the ovule is covered by this is the outer integument and this is the inner integument. So the testa is derived from the outer integument and tegmen is derived from the inner integument of the ovule. So these are some of the features which are very, very important. The difference between the testa and tegmen is also very important. Okay, so make sure you note it down carefully. The second part of the seed is the cotyledon. We know that the cotyledon is the swollen structure. It is the swollen structure. And what is the function of cotyledon? It helps in the storage of food. Okay, so if we see this diagram, so the innermost part that is the swollen in nature, that is called the cotyledon. Okay, so it helps in the storage of food. Okay, and the third main part of the seed is the embryo axis. We know this is the embryo axis. Okay, so These three parts of the seeds are very important, so make sure you note it down carefully. Now, if we see the structure of the seed in some of the seeds like black pepper, these examples are very important, okay? Black pepper, beetroot. In these kind of seeds, the remnants of the nucellus are also persistent, means the residual persistent nucellus is present that form the perisperm. Okay, so in sometimes perisperm, this term is very important. Perisperm is the nutritive, if this is the seed and the outermost covering is the testa and the inner one is the tegmen. So inner to tegmen there is a nutritive layer of the nutritive tissues which forms or which surrounds the seed, that is called the perisperm. Sometimes what happened, in black pepper and the beetroot, the nucellus part is residual. Okay, so the residual persistent nucellus part of the ovule, where nucellus is present in the ovule. So in the seed, in the maturation of the seed, the residual part will persist of the nucellus that form the special kind of nutritive layer around the seed, that is called the perisperm. So this example is very important. What is perisperm? And perisperm is made up from the nucellus part. This part is important. Okay. So now if we see in the mature seed, if we see the mature seed, it may be divided into two categories. So look at this picture. So mature seed is divided into the two categories. First one is the albuminous seed and second one is the non-albuminous seeds. Okay? What do you mean by albuminous seeds? Albuminous seeds are the seeds when the endosperm is completely consumed by during embryo development. Okay, so albuminous seeds are the seeds when the endosperm is completely consumed during the embryo development. Why they are completely consumed? Because endosperm serves the food during the embryo development and no residual endosperm left. No residual endosperm is left. That kind of seed is called the albuminous seeds. Okay. When the endosperm is completely consumed during the embryo development, no residue left, that kind of seed is called the albuminous seeds. So the examples of the albuminous seeds are the pea, groundnut, etc. So opposite to albuminous seeds, the non-albuminous seeds or the X-albuminous seeds. Another name of the non-albuminous seeds is the X-albuminous seeds. So here some part of the endosperm retain during the embryo development. Okay. So some part of the endosperm left, some residues are left during the embryo development. Residues are left during the embryo development, those kind of seeds are called the X-albuminous or non-albuminous seeds. Okay, I hope the difference is clear to you all. So the example of the X-albuminous or non-albuminous seeds is wheat, maize, barley, and castor. Okay, so these all examples are very, very important, that in which plant albuminous or the non-albuminous seeds are present. I hope this difference is clear to you all. Now, when the seed gets mature, when the seeds gets matured, what happened? Okay, so when the seeds gets matured, the water gets decreased from the seed. So the level of the water inside the seed gets decreased. Okay, when the water gets decreased in the seed become dry. Okay, when seed become dry, it will slow down the metabolic activity inside the seed. Okay, when it is metabolically inactive, so the embryo is also inactive in nature. That state of inactivity is called the dormancy. This is very important. What is dormancy? Dormancy, when the seed gets matured, the level of water gets decreased and the seed becomes dry. The activity metabolically, they are inactive. The embryo is also inactive in nature. That state of inactivity is called the dormancy. Okay, so dormancy. So this term is very, very important. What happened during the dormancy condition? These conditions are very important. The level of water, inactivity, dryness, embryo gets inactive and the seed becomes dormancy in nature. So if we see the structure or the section of the seed, if we see the structure of the section of the seed, this is the maize seed. If we see the structure, so maize seed, if we see this part, we have discussed in the previous class these all terms. I hope these all terms are clear to you all. So in the seed part, we know that in the hypocotyle, that is the below the level of the cotyledon, the radicle is formed. The radicle forms the root system, and the sheath of the radicle is called the coleorhiza, that we have discussed in the previous class. Sheath of a radicle is the coleorhiza. So plumule forms the shoot system. Okay, so the covering or the sheath of the plumule is the coleoptile. I hope these all terms are clear to you all. And the scutellum, which contains the cotyledon. Okay, so where the scutellum is present, in which type of the embryo the scutellum is present? So we have discussed the scutellum is present in the monocote embryo. So this is the structure of monocote seed. Okay, monocote. Why monocote? Because scutellum contains a single cotyledon. That's why it comes under the monocote embryo. Okay, so this is the structure of the this is the structure of the maize seed. This structure is also very important. Sometimes in your board exams, this structure is given and the labeling is not mentioned, and they asked you about find out the each part of this structure. So this kind of questions have also asked in your previous board exams. So these all parts are very, very important. Make sure you practice this diagram and understand each and every part of it. Now, the last part of under the post fertilization events is the fruit. Now fruit, if we talk about the fruit, that fruit is formed from the maturation of ovary. Okay, so seed is formed from ovule, and fruit is formed from the maturation of ovary. Okay, so there in the seed part we have discussed the different layers that like testa, tegma, and the perisperm. So the fruit wall is called the pericarp. This is very important. The fruit wall is called the pericarp. The wall of the fruit is called the pericarp. Now, there are two kinds of the fruit. Present, fleshy fruit and dry fruit. Fleshy fruit, as the name suggests, the fruits that contains the pulpy in nature, which contains pulp inside it. So we have example like guava, orange, mango. These all comes under the fleshy fruits, and dry fruits are like groundnut and mustard. Okay, so these examples are important. Now, if we see the fruit types, there are two kinds of fruit present. First is the false fruit, second is the true fruit. This difference between the true fruit and the false fruit is very important for your board exam point of view. So, what is false fruit? False fruit and the true fruit. First, we will discuss about the true fruit. True fruit means the fruit which only and only develop from the ovary. We know that the fruit is made up of when the ovary gets mature. So, the true fruit is the fruit which only develop from the ovary. That kind of fruit is called the true fruit. So, under the true fruit, there are examples like mango, grapes, fig, etc. In case of the false fruit means the fruit which does not derive from the ovary, but they are developed from the thalamus or other floral parts. Okay, so the false, that's why it is called the false fruit. Okay, so it do not develop from the ovary, but it develops from the thalamus. This is the floral part, or other different floral parts. So these kind of fruits are called the false fruit. The example of the false fruit is very important, like apple and the strawberry. These are the examples of the false fruit. If you see the diagram of the false fruit, this is the apple and this is the strawberry, which comes under the false fruit. If you see here, this is the seed, and this part, the eatable part of the apple and the strawberry, is derived from the thalamus. See here, this is the thalamus. So, this is not derived from ovary, but it is derived from the thalamus. That's why the apple and the strawberry comes under the false fruit. I hope this difference is clear to you all. The examples are also very important. So next type of fruit is, the third type of fruit is the parthenocarpic fruit. Okay, the parthenocarpic fruits are the fruits which develop without fertilization. Okay, so this point is very important. The parthenocarpic fruits are the fruits which develops without fertilization. The fertilization not happens during the parthenocarpic fruit, formation of the parthenocarpic fruit. So when the fertilization not happen, then the seed is not formed. So these kind of parthenocarpic fruits are the seedless fruits. We can say this. Okay. So the parthenocarpic fruits are the fruits which develop from the without the fertilization, and they are mostly seedless fruits. Sometimes they are induced by the application of growth hormones. Okay, so we know that there are multiple growth hormones present in the plant. So with the activity of the growth hormones, the parthenocarpic fruits are developed. Okay, so in this, a very important example is there, that is the banana fruit, which is a parthenocarpic fruit and formed or developed without the fertilization. That's why this is the seedless fruit. Okay, so example is also very important. So I hope these all terms are clear to you all. Now, if we talk about the advantages of the seeds, a very important advantage is the seeds of the large number of the species live for the several years. So, in some species, the seeds of the from the several years, they live and used by many scientists. Okay, so if we there are the two examples in the oldest and the recent. In the oldest record, there is an example of Lupinus arcticus. This is the plant. Okay, lupine is the common name, and the scientific name of this plant is the Lupinus arcticus. So this is the plant which is excavated from the Arctic tundra, and the findings under this plant is the seed germinate and flowering after 10,000 years of the dormancy. So after the 10,000 years of the dormancy, the seeds of this plant germinate and flowering. Okay, so in the recent record, there is a date palm. This is a tree, date palm. The scientific name of the date palm is the Phoenix dactylifera. Okay, its scientific names are important. So they are excavated from the King Herod's palace, which is near the Dead Sea, and they have the viable seeds. They have the viable seeds which are 2,000 years old. Okay, so these two examples are important. So make sure you note it down, and their advantages of the seeds are also important. So this is all about the post-fertilization events. We have discussed each and every part of it. We have discussed about the endosperm, okay, the structure of the endosperm. Second, the embryo, monocot embryo, dicot embryo, and its structure. Third is the seed, how seeds form, the structure of the seeds, different layers of the seeds, different types of the seeds, and the last is the fruit. We have discussed the different types of the fruits, parthenocarpic, true fruit, false fruit, and the advantages of the seed. So I hope this lecture is beneficial to you all. This is all about today's lecture. Keep studying, study hard, study smart. If you like this lecture, like, share, and subscribe my channel, that is PrepBiology. Thank you so much.